The fourth type of membrane receptors is G-protein coupled receptors, which reside in the membrane, and they constitute a large family of receptors. An example of these receptors are the beta-adrenergic receptor, the glucagon receptor, and the alpha-1 adrenergic receptor. All of these G-protein coupled receptors transduce their intracellular effects through activation of specific G proteins. Importantly, the activation of these G proteins results in the activation of a specific effector enzyme. The activation of these effector enzymes results in the production of unique and specific intracellular second messengers inside the cytoplasm.
One of the key things about G-protein coupled receptors is when a G-protein coupled receptor is activated, it will invariably produce an intracellular second messenger. Okay?
Cyclic AMP is an example of a second messenger. Before receptor activation, this molecule would have been absent inside the cytoplasm. After receptor activation, this molecule concentration goes up, and that's why we call that a second messenger. Second important feature about second messengers is that they are typically small organic molecules. Okay? They are not proteins in nature. They are not peptides. They are typically organic small molecules. Here they are. Here are the examples. Cyclic AMP is one, IP3, inositol trisphosphate, diacylglycerol, calcium, cyclic GMP, nitric oxide. Second messengers are typically small molecules that are not present during the resting state of a cell.
Activation of either the beta-adrenergic receptor by epinephrine or the glucagon receptor by glucagon activates GS-alpha, which in turn activates adenylate cyclase that produces the second messenger cyclic AMP. G-protein coupled receptors are highly specific. Only epinephrine can activate the beta-adrenergic receptor, and only glucagon can activate the glucagon receptor. Activation of the alpha-1 adrenergic receptor by epinephrine results in the activation of the trimeric G protein designated GQ. GQ dissociates to produce GQ alpha bound to GTP, which then can activate phospholipase C. Phospholipase C will hydrolyze PIP2 in the membrane to produce diacylglycerol DG in the membrane and IP3 in the cytoplasm. IP3 moves to the ER and releases calcium from calcium stores in the ER. Ultimately, 3 second messengers are produced in response to receptor activation by epinephrine. Activation of the beta-1 adrenergic receptor by epinephrine or norepinephrine activates GS-alpha, which activates adenylate cyclase and produces cyclic AMP as a second messenger. Activation of the glucagon receptor by glucagon activates GS-alpha and activates adenylate cyclase to produce cyclic AMP as a second messenger. activation of the alpha-1 adrenergic receptor by epinephrine or norepinephrine activates GQ-alpha to activate phospholipase C and produce IP3DG and calcium as second messengers. If you like this video, please subscribe. Thank you.